give you variation. In bacteria, you don't have mitosis because you don't have a membrane-bound nucleus. You generally have a single large circular piece of DNA. You might have a few smaller pieces of DNA as well called plasmids, but here's the single circular chromosome. It has an origin of replication and a terminus of replication. So you start replicating the DNA from the origin, and then you end at the terminus, and you end up with 2 copies of DNA. And the 2 copies will be attached to something along the length of the plasma membrane as the cell then elongates and divides. So you end up with 2 genetically identical cells. This is not something we're going to focus on. So cell division in bacteria is not our focus. We're going to look at what happens during mitosis and cytokinesis in eukaryotes. But just so you know that prokaryotes do it their own way, and it's different than what we do, I had this figure in here. All right, so most of the cell divisions that happen in your bodies are through mitosis and cytokinesis. So you're making genetically identical copies of each of the cells. And then there's meiosis, which gives you variation because you are not making genetically identical copies of each cell. And during meiosis, you're going to make cells that have half as many chromosomes as the original cell. During mitosis, you make the same exact number of, the same exact copies, which means the same number of chromosomes, identical DNA, unless there was a mistake in replication. And we will talk about mistakes in replication a little bit. You're also reading a section of one of the modules in the textbook about cancer, which will deal with mistakes in cell division. And if you're interested in cancer biology, there is a lot more in your textbook. And we do have an upper level course that focuses just on cancer biology. It's Bio 425. So after, so you need Bio 101, 102, 206, 229, then you can take cancer bio.
Nice. You want some?
But we're not going to focus on that except for the little bit you're going to read in your textbook, which is habits. So most of your cells are that are dividing cells are going through a regular cycle where most of the time of the cell is spent, about 95% of the time of the cell is spent in the interphase period where the cell is going through basic biochemistry and maybe growing, and only less than 5% of the time of the cell is actually going through nuclear division followed by cell division. Cells can be arrested, so that means they're not going to go through the whole cycle. So there are some cells that will never divide. We used to think that your brain cells were like that, and now recent evidence suggests that they actually aren't, and they actually do divide. They just don't do it very often, and if you stimulate them, in some cases, they can divide. So that's an interesting area of neurobiology these days, looking at how neurons that used to be thought not to be dividing are actually dividing. So here's my diagram of a cell. First, you have nuclear division. Then you have cytoplasmic division, cytokinesis, and then the cells grow back to the original size of the parents. And that's usually what happens in a regular cell that's going through the regular cell cycle. There are special cases where you don't get cell growth after cell division. And that happens during part of embryogenesis also, when your original egg cell that was fertilized just keeps dividing without growing, and then you get growth after that. So our focus today is going to be the mitotic phase, not so much the interphase, although we should know basically, very basically what happens during interphase. So the mitotic phase includes both mitosis and cytokinesis. So mitosis is referring to the division of the chromosomes that leads to karyokinesis, which is the division of the nucleus. And then you don't have to know karyokinesis, although it's on the list of things to look up. So you do look it up. The cytokinesis is the division of the cytoplasm. Now, one of the interesting things that we will do towards the end of the semester is during interphase, you have to replicate your DNA. And that includes all of the chromosomes, as well as if you are a bacterium, the plasmids have to be divided. If you're dealing with a eukaryote that has multiple organelles like chloroplast and mitochondria, they have their own DNA, and then you have to make copies of those mitochondria and chloroplast. And how do they divide? How do mitochondria and chloroplast divide? How do mitochondria and chloroplast divide? Like bacteria. So they do their own division. And then when the cell goes through cytokinesis, half of the mitochondria and chloroplasts go to one side, and half of them go to the other side. So you have to have coordination of what happens with the mitochondria and chloroplast with what happens during the cell cycle. Right. So for now, DNA replicates. And we're not going to talk about the details of how that happens towards the end of the semester. Now, you do have to know that DNA is packaged into chromosomes, and chromosomes in eukaryotes have DNA wrapped around. What are your chromosomes made of?
Protein.
DNA plus, not RNA. DNA is made up of nucleotides, but what is it in your chromosome? You have DNA, and what else? What other macromolecule do you have in your chromosomes? When the chromosomes are condensed like this, that DNA is wrapped around what? Histones. What are histones? Proteins. They're proteins. So your chromosomes are DNA wrapped around proteins. So when you stain chromosomes to do a karyotype like this, you're looking at chromosomes from a dividing cell. When the cell is going through the majority of the cell cycle, the chromosomes do not look like this. This only happens during the mitotic phase. So if you happen to catch a cell that happens to be in the mitotic phase, and then you stain its chromosomes, they will look like this. The 95% of the time that the dividing cell is in interphase, the chromosomes do not look like this. And so when you stain them, you will not see anything like this, and that will be useless in terms of doing a karyotype. So when we do a karyotype, we stain the chromosomes, we look to see if you have the right number of chromosomes. You can check to see whether you have the X and the Y in humans. In this case, you have a copy of an X chromosome and a copy of the Y chromosome, so this individual happens to be, one would hope, a male. Doesn't have to be. You could have XY females. This, we hope, is an XY male. You could also have XX males. So chromosomally, this person would be an XY, potentially male. And all the other chromosomes, you look to see, all right, so you have a bunch of chromosomes, you count to make sure you have the 46, sometimes you have 47, if you have 47, what does that tell you? Usually that means Down syndrome, doesn't have to, because Down syndrome is the most common trisomy, but you could have other trisomies that are usually fatal. And you might look to see whether there's a piece of a chromosome missing, for example. So if an arm of chromosome 5 is missing, then you could have one type of syndrome called Cri-du-Chat, cry of the cat. If you have other pieces missing, they may not survive. So most of the time, if you are a woman and potentially getting pregnant, you will have a spontaneous abortion and not realize that you were even pregnant because the embryo gets rejected because there are too many problems with it. Usually means missing chromosomes, broken chromosomes, etc. So most of the time, you don't even realize that you had a pregnancy because it's immediately rejected by the body. Sometimes you have a miscarriage, which means you realized you were pregnant, and then the pregnancy was terminated by your body. And so then you have a miscarriage. But most of your miscarriages happen before you realize you were even pregnant. So this happens to be a normal-looking group. But one of the things that you should know is that when chromosomes are condensed like this, they are already replicated. So each of these chromosomes has 2 copies attached to each other. So this is not 46 normal chromosomes. These are 46 duplicated chromosomes, ready to go through mitosis. So, you always talk about the 2n number for typical diploid species. So what does that mean? That means that your somatic cells, those are your non-reproductive cells, have 2 copies of each type of chromosome. So in humans, like yourselves, when we talk about 46 chromosomes, that's your 2n number. Which means your n number is 23. That means you have 23 different types of chromosomes, and you have 2 copies of each of them, making 46. So here, when you have 46, there are 23 pairs. There are 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, etc. And the 23rd pair is the X and the Y. And you could be XX or an XY if you're normal. And you could be XO or XXX or XXY or XYY and all those abnormalities, which we will talk about when we do genetics. So normally, your 2n number is 46, and your n number, which will be the number of chromosomes that your sperm and egg will carry, is 23. So as your cells in your reproductive cells, the germline, go through meiosis, they will take 46 chromosomes and they'll divide them up so that each egg or sperm gets only 23, one of each version of the chromosomes. One thing to remember is that here you have 46 chromosomes, but each one of them is duplicated. So for chromosome 1, you have 2 chromosome 1s, and each one of them is duplicated. So you actually have 4 copies of chromosome 1, and each pair of them are joined together. And those copies that are joined together are called chromatids. So you have to know the term chromatid. In a given replicated chromosome, there are 2 sister chromatids joined at the centromere. And during mitosis, you are going to separate the sister chromatids so that you're back to your normal 2n46, and each of those chromosomes is only 1 copy. So we're going to take a look at the details of all of that. But before we do that, start at the top. Here's the condensed chromosome. At the bottom, you have your DNA. If you were to stretch the DNA in your chromosomes, it will be several meters long. And somehow, all of that DNA is packaged inside the tiny nucleus of your tiny cells. So 250,000 times more condensed than if it were just stretching. Yes, yes, ma'am. I'm sorry, say that again? So in the chromosome at the top here. This is one chromosome. The chromosomes are not joined. So this is chromosome 1, let's say. And there's another one looking like this, which is the second copy of chromosome 1. And then you have another pair, chromosome 2, another pair, chromosome 3. So those chromosomes are not joined together. The DNA was never joined together. Your chromosomal DNAs are separate from each other. Each of these chromosomes, this chromosome is not joined to that chromosome is not joined to this chromosome. So you have 46 molecules of DNA, each one of them making a chromosome. And in each chromosome, the DNA is wrapped around histones, the proteins that one of you mentioned. So that's the first level of condensation that you get, is the wrapping around the histones. And then the histones are looped like this, so that gives you the chromatin fiber. And that's the normal status of the chromosome when it's in interphase. And then you get these looped domains where this is looping on top of itself. And then the loop domains are looped on top of themselves. So you get multiple layers of looping until you get to the condensed chromosome. The condensed chromosome is not going to be used to make any proteins. So any condensed region of the chromosome is not open to transcription enzymes. And during interphase, when you do get transcription and translation, some parts of the chromosome remain condensed, and you don't use those genes. And other parts of the chromosome unfold like that, and you use those genes. And how you condense or decondense the chromosomes is the subject of a relatively new field of biology called epigenetics. And at the very end of the semester, you are reading an article about epigenetics, and we're going to talk about it in class. So how you get to the condensed versus the decondensed forms to regulate gene expression is a very interesting new topic, relatively new topic. But as far as we're concerned, during mitosis, you need to condense the chromosomes so that you can move them around, because you're going to divide the chromosomes between cells. So you can't do it if they're open like this, because they get tangled up. But if you condense them, then these can be moved around. So wrap them around his